A drug is a chemical that affects physiological function. Drugs exert their action by binding to a specific target molecule, usually a protein, thereby modifying its function. This may switch the target on or off and affects biological activity of the target and physiological function. There are four principal targets for a drug to act on, which are as follows: receptors, ion channels, carriers, and enzymes. A receptor is a protein molecule found in the body that acts as a recognition site for the body's natural mediators, or ligands. A ligand will specifically bind to the receptor, which then transmits a signal and elicits a biological effect. A ligand can switch this response on or off. Drugs that act on receptors often resemble the ligand structurally and bind to the receptor. Some drugs stimulate the biological effect, while others inhibit it. Salbutamol is an example of a drug that acts on adrenergic receptors, whose natural ligand is adrenaline. Now select a drug and see what happens. Ion channels are proteins in the membrane that form a pore or channel through which ions can pass, like a plug in a sink. Ion channels only allow specific ions to pass through them, and only when activated. Drugs can bind to the ion channel, thereby altering its function. Local anesthetics are an example of drugs that block sodium ion channels. Carriers are membrane-bound proteins that transport specific molecules, for example ions, and small organic molecules such as glucose, across the plasma membrane of a cell. The molecules bind to specific recognition sites on the carrier. This induces it to change, and in doing so, the molecule is transferred to the other side of the membrane. There are many types of such carriers, including sodium-potassium pumps, and uptake of neurotransmitters. Some drugs will target the recognition site and thus block the transport system. For example, cocaine acts by blocking the uptake of noradrenaline. Enzymes are biological catalysts that speed up chemical reactions without being changed themselves. There are more than 500 different enzymes in every cell, and without enzymes, reactions would be too slow for life to continue. An enzyme works by binding the substrates in the active or catalytic site in such a way that chemical reactions are induced and products are formed. Approximately half of all prescribed drugs act by inhibiting the action of specific enzymes. An example of a drug that targets and inhibits enzymes is aspirin.
Drugs that act on receptors often resemble the natural or endogenous ligand structurally and have an affinity for specific receptors. Drugs that bind to the receptor and initiate a biological response are called agonists and are said to have an intrinsic activity. Conversely, antagonists have similar affinity for the receptor but do not initiate a biological response and hence have no intrinsic activity. These drugs will block or reduce the response of the natural ligands. Now add a new drug and state whether it is an agonist or antagonist. Receptor action can occur either very rapidly, for example milliseconds during nerve transmission, or over days, for example steroid hormone receptors. This depends on the type of receptor and its cellular localization. There are 4 main receptor types, or superfamilies. Firstly, the ligand-gated ion channels, or ionotropic receptors, which are membrane-bound and directly coupled to an ion channel. The binding of the ligand to the receptor results in the ion channel opening and allowing ions to enter the cell and elicit a biological response. These receptors are fast and important in neurotransmission. For example, when acetylcholine binds to nicotinic acetylcholine receptors, causing sodium ions to enter the cell. Nicotine is an agonist for this receptor and elicits the same effect. The second type is the G-protein coupled receptor, or metabotropic receptor. This is located in the membrane. When a ligand binds to this type of receptor, a signaling cascade is initiated via a heterotrimeric protein called a G protein. This is also called a second messenger system. The response might be stimulatory or inhibitory, depending on the G protein type. While not as fast as ligand-gated ion channels, these receptors still produce a response within seconds. This is analogous to the Olympic torch, where the torch is first lit by a match, the ligand, then passed from person to person, the second messengers, until it lights the Olympic flame, the biological effect. An example, adrenaline, which stimulates the fight or flight response, binds to metabotropic adrenergic receptors. Drugs called beta blockers, such as propranolol, inhibit this and reduce the effect of adrenaline. The third class are kinase-linked receptors. These membrane receptors contain enzymatic activity called kinase, which transfers a phosphate group between amino acids in different proteins, phosphorylation. This then activates a signaling pathway, culminating in a cellular effect, such as an increased cell metabolism. This second messenger system is like a series of dominoes, where the ligand knocks over the first domino, which then knocks over the next one, and so on until the finale. These receptors are relatively slow and take several hours to produce a biological effect. The insulin receptor is an example of this receptor type. Finally, nuclear receptors, which are found in the cytoplasm and/or nucleus, and regulate gene transcription, i.e., they switch genes on or off. The steroid hormone receptors are an example of a nuclear receptor. Drugs which act on nuclear receptors take longest to exert their action, requiring hours or even days to exert a biological effect.
Carriers are cellular proteins which span the plasma membrane and act to transport drugs and other molecules into and out of cells. The actions of these proteins may be compared to revolving doors in a building. A carrier binds a molecule on one side of the cell membrane and transports it to the other side by changing its own shape. The molecule can only be transported if it fits the binding site of the protein carrier. Carriers can be subdivided into those which require energy, known as energy dependent, and those which do not, known as energy independent. Energy independent carrier molecules can be further subdivided into transporters, symporters, and antiporters. Transporters carry a single molecule in one direction across the membrane. Symporters carry two different molecules in the same direction across the membrane, while antiporters transport two molecules in opposite directions across the membrane. These energy-independent carrier molecules can only operate down a concentration gradient. Energy-dependent carriers can transport molecules across membranes against the concentration gradient, but require energy in the form of ATP for this function. Drugs can act to modulate the function of carrier molecules directly or indirectly. Some drugs bind competitively to the carrier molecule, preventing binding of the endogenous ligand. An example of this is probenecid, which binds competitively to the acidic carrier in the renal tubule, preventing the binding and hence excretion of penicillin. Indeed, probenecid used to be given with penicillin in order to prolong its action. Indirect or allosteric inhibition of carrier molecules occurs when a drug binds to a separate site on the carrier and alters the three-dimensional structure of the binding site, thus preventing the binding of endogenous ligands. Examples of this type of inhibition are selective serotonin reuptake inhibitors, for example citalopram, cardiac glycosides, for example digoxin, and proton pump inhibitors, for example omeprazole. Ions are unable to cross the cell membrane without the help of proteins, for example ion channels, which span the membrane. Ion channels can be compared to a doorway into a building. Rather than binding the transported molecule, they form a channel through the membrane. These channels are small pores that permit water or particular ions such as sodium to pass through. Ion channels can exist in an open or closed form, with the open form allowing the movement of ions and the closed form preventing this movement. Ion channels are often selective in terms of the ions they allow across the membrane. For example, sodium channels will only permit the passage of sodium ions, and potassium channels will only permit the passage of potassium. Some ion channels are activated by the binding of endogenous ligands to a specific receptor which forms part of the ion channel. These are called ligand-gated ion channels. Other ion channels are activated following a change in voltage and are known as voltage-gated ion channels. Ion channels can be inhibited by drugs which directly block the ion channel and prevent ion movement. An example of a drug which acts in this way would be the local anesthetic lidocaine, which specifically inhibits sodium channels. Ion channels can also be modulated indirectly. In other words, the binding of a drug to a site on the associated receptor can either increase or decrease the likelihood of the channel being open. For example, rocuronium, which competes with acetylcholine for binding to the nicotinic acetylcholine receptor, prevents the passage of sodium and potassium ions across the membrane. On the other hand, the binding of benzodiazepines to a separate site on the GABA receptor increases the movement of chloride ions.